Hello class and welcome back. Last period we discussed about the nutrition in plants. So today we are going to discuss about a side of photosynthesis, that is chloroplast. And chloroplast is a plastid, like a cell organelles. These cell organelles, plastids, under the plastids you will find chloroplast, leucoplast, okay, and chromoplast. So this in the plastids, the main thing is chloroplast, which is the main site for the chemical reaction that takes place. Everything, okay, like it's like a factory for the plants. Chloroplast is like a factory for the plants, food factory. So the food is made by the chloroplast. The food is made by the chloroplast. So let's see the chemical reactions during photosynthesis placed in chloroplast. Chloroplasts are plastids which are plant cell organelles 3 micron to 6 micron in diameter. So chloroplasts are the site where chemical reaction, that is photosynthesis, takes place, and chloroplasts are plastids, okay, which ranges from 3 micron to 6 micron in diameter. Now, how can we find this chloroplast? In chloroplast, you take a leaf. Okay, you take a leaf, and so this is your leaf, right? You cut this side. Okay, you cut this side in a thin slide, with a blade. After cutting, you can observe it. Wash it and you can observe it under the microscope. Then you will find the chloroplast. So this called as palisade mesophyll cells. So you look at the figure 1.7, the structure of leaf showing chloroplast. You have xylem, phloem, that is vascular bundles, cuticle, layer covering the cell walls, upper epidermis, palisade cells, spongy mesophyll, single palisade cell, then cellulose wall, okay, cytoplasm, cell vacuoles, nucleus. So below that you will find stroma as well as guard cell. Okay, so everything is take place by the chloroplast. Either it is meant by transporting the food or transporting the plants. Okay, it takes place in the chloroplast. So making the food as well as transporting also responsible by the chloroplast. Now usually chloroplast can be seen easily under a light microscope, but finer details of organelle can be observed only under an electron microscope. You see that in your school, right? In our school, that is the light microscope that you see. Okay, it's very not good for the further studies, like into and detail. They will only find, or they will see only what they will show you only small details, not the full organelles. Okay, but electron microscope means you put a wire, or you put with the help of the electric, okay, you can find the good organelles. or everything, including the stoma or gut cells. Okay, cells. So a typical palisade cell of green leaf may contain 100 or more tiny chloroplasts in it, and a whole leaf may contain thousands of palisade cells. In a single leaf, it contains 100 or 1000 chloroplasts. Understood? It does not contain one chloroplast in every leaf. So you see, in one tree you'll find how many leaves? Maybe 1,000, right? Maybe 1,000 or 10,000. So in that 10,000, you'll find in one leaf you'll find 100 or 1,000 chloroplasts. The spongy mesophyll cells vary in shape and fit loosely together, leaving a lot of space between them. So this spongy mesophyll there slowly starts to depart away. Okay. The functions of the palisade cell, and to a lesser extent of the spongy mesophyll, is to make food by photosynthesis. So this palisade cell, they are the responsible factor for making the foods in the chloroplast. And when photosynthesis is rapid, the mesophyll cells use up carbon dioxide. As a result, the concentration of carbon dioxide in the air space falls, and more carbon dioxide diffuses in from the outside air through stomata and reaches the chloroplast. So when the photosynthesis, or when the sunlight is too much, or photosynthesis is too much, this mesophyll cell starts making the food rapidly. So when you go fast, right, you need more energy. So you need more energy. So this mesophyll cell takes the carbon dioxide, takes your carbon dioxide in the cell. Then that carbon dioxide, when they are used up, right, they will start taking from the other environment. Okay, so that time the carbon dioxide or the air present in the outside of the plant or outside of the cell become little bit disturbed. Okay, this diffusion continues through the airspace up to the cell which are using carbon dioxide. Water is carried by xylem vessel to the chloroplast. There is a tiny thin waxy layer called the cuticles over the epidermis, which helps to reduce waters. Now you have heard the term diffusion and osmosis, right? Osmosis means transferring of water. Diffusion means transferring of gases. So when the mesophyll cells makes more energy or makes more food, they require more energy and they take the food. Okay, they take the carbon dioxide in the cell. So during that time, diffusion occurs. During the time diffusion occurs, they start exchanging the gases. Since they have used up inside the cell, they will take from the higher concentration, from the outside concentration to the lower concentration of the cell. So that is when the diffusion takes place. Okay, and and the osmosis or water vapor, right? Transferring of water vapor occurs, but there is no loss of water vapor in the cell. Why? Because there is a cuticle, which is a thin layer covering the cell wall, right? This thin layer protects the water or protects the cell, allows the waters to stay inside, not go outside of the cell, like evaporation. Okay, evaporation does not occur too much. So because of the cuticle, the waters are safe, and xylems are responsible for the transferring of water molecule. So that's all about the chloroplast side of photosynthesis. Next class we will discuss about the role of photosynthesis, and please make a fair copy of biology and write your copy in a proper manner. Thank you.